Hello and welcome to this presentation on genetically modified foods. In this unit, we're not only going to be learning about what genetically modified foods are and the different types and how they're produced, but we're also going to be analyzing food samples that you bring in to find out if the foods that you're eating are genetically modified. Um, so let's take a look. A little bit of history to start. We've been genetically modifying our plants and animals actually for thousands and thousands of years. The way that that has worked is through a process called artificial selection, also known as selective breeding. What that means is that you're taking the two plants or the two animals that you feel have desirable traits and letting them reproduce, hoping that the offspring have both of those traits. For example, if you have a cow that produces lots of milk and a bull that is very healthy, if you let the two reproduce, hopefully the offspring will be high milk producing and healthy cows. Of course, genetically, you don't always know exactly what you're going to get. Certainly, you'd have a better chance if you let those two reproduce versus the sick and low milk producing animals and hoping that the next generation is better. But at the same time, that's the way it's been done. And so, again, we've been genetically altering our plants and animals for thousands of years using this artificial selection or selective breeding process. In the case of genetic engineering, though, we've been able to change the process a bit. And so there are some terms to be familiar with, some of which you already know. Transformations, for example, moving DNA from one cell to another, you are already familiar with ways to do that. Recombinant DNA is a term that means DNA combined from two or more sources. So for example, the bacteria that you produced that had the green fluorescent protein gene or the bioluminescence gene, those were bacteria that were recombinant because they had the DNA in them from jellyfish or from those marine organisms. Transgenic actually technically would be the bacteria themselves. The DNA would be the plasmid, the recombinant DNA. But transgenic organisms transformed with recombinant DNA. And a cell that has been transformed with recombinant DNA is considered to be transgenic. GMOs are transgenic organisms. An organism that is genetically modified means that it has been altered in a way that does not occur naturally by mating and or selective breeding. And so you can change the traits of food and livestock through selective breeding, but we do not consider that to be genetically modified, although the traits might be different. With GM or GMO or GE, we're talking about things that cannot happen naturally. For example, being able to take genes from fish and put that into plants doesn't make a difference how much romantic music you play, the plants and the fish aren't going to reproduce. So we can cross the species barrier using genetic engineering. I'm going to have you take a look at a video here. You're going to find the link in the calendar. And this video is going to describe genetically modified papaya. On the left, you see papaya grown in Hawaii that are infected with a virus called papaya ring spot virus. And this virus was decimating the papaya crops in Hawaii about 20 years or so ago. And the plants on the left are contaminated with that virus, infected with that virus, and the trees are stunted. The fruit is no good to sell. This was having a major problem within the papaya industry in Hawaii. The plants on the right are genetically modified papaya to be able to resist that virus. They are exposed to the virus. They're growing just fine. And while the video is going to describe how this worked, I know papaya might not necessarily be high on your list of things that you consume on a daily basis or even a monthly or yearly basis. But you can imagine in Hawaii this was a big deal. This virus was decimating their crops, and if the same were happening to corn or soybeans here in Wisconsin, this would be a major deal. So I want you to pause this video here for a second. I want you to go link into the other video, watch that video, take notes, and then I'll come back and describe a little bit more about this. So again, pause this, watch the other video. Based on that video, what you saw were 4 steps about how crops are genetically altered. The first step they mentioned was to determine whether genetic engineering is the most effective way to solve a plant's problem. In this case, being able to genetically alter the crops meant that they would be able to recover that industry. But they had to find a solution. And the solution involved being able to make the plants resistant to that virus. And so you might be able to do that through selective breeding, through being able to move the plant from one island to the next. They tried those methods, didn't exactly work. Part of the problem here with selective breeding is that while it's always an option, it can take decades and multiple generations to get the desired trait. That's a long time, and they didn't have that kind of time in this case. In addition, it involves randomly mixing genes, yielding random results. You never know exactly what you're going to get. Now, genetically, we can set up our Punnett squares. We know the chances of one trait versus another. But remember, our Punnett squares that we did in biology were taking a look at one, maybe two traits. We're talking about thousands upon thousands of genes, some of which are recessive. And so you never know exactly what you're going to get in the next generation. You might be getting some surprising traits that you never anticipated. And it takes a lot of trial and error. That takes time. In genetic engineering, we know exactly which gene we're moving, and we can study the effect of that specific gene on the next generation. In some ways, a much more controlled experiment. You have to identify the gene. In this case, what they did was they identified that there's a gene that they could take from the virus itself and put that into the papaya. By doing this, the plant would become resistant, almost like giving it a vaccination. Once they identify the gene, then they have to take that gene from the donor organism, in this case the virus, and transplant that gene into the plant's DNA, cutting and pasting the two together, a process that we're going to be doing later this semester. We also know that we have to move that gene into the plants, and you know how to do that with heat shock transformation, or as the video described, using gene guns, a high-speed particle bombardment method, a gene gun, being able to move the DNA into the plants. Once you have that, now we have to actually take those genetically altered plants and plant them and test them, see if they actually work. Do this, of course, in a controlled environment first. You don't want to just release virus out there into the environment and see, okay, well, how does this affect the plants that we just produce? So you got to start in the greenhouse. In doing so, there's a lot of testing that would happen. So while it takes time to produce the genetically altered crop, it also takes time to test to make sure that it's doing what you want it to do, in this case, be resistant, but also still fit the criteria set forth by the FDA, the USDA, and the EPA. The FDA is the Food and Drug Administration. They're going to make sure that the food we are planting and growing is safe to eat. Also, the USDA, the U.S. Department of Agriculture, is going to make sure the food is safe to grow. And then also the EPA, the Environmental Protection Agency, is going to help make sure that the food is safe for the environment. You got to satisfy all three of these groups. And that's true for any genetically modified crop. Not necessarily completely true for every selectively bred one, but definitely true for every genetically modified crop. In this case, what they found was that there are 125,000 pounds of marketable fruit per acre per year, 25 times the amount produced by non-modified fruit. And so a major improvement over where they were before with fruit that was infected with the virus. Now, in the continental United States, the two major common genetically modified traits are not papaya ring spot virus, being able to withstand that virus, but instead two other traits, one called HT. HT stands for herbicide tolerance. And the H, in this case, an herbicide, these are chemicals that kill plants. They don't just kill weeds, they kill plants. Roundup is an example of this. And if you've ever used Roundup to kill weeds in your sidewalks or in your driveway, or to clear out an area, you know it's very effective. If you use it to try to kill the dandelions in your grass, you're going to not only kill the dandelions, but you're going to kill the grass along with it. Herbicides. In this case, you're taking a look at a field that is soybeans. And these soybeans, as you know, I'm sure you know, here in Wisconsin, we rotate our crops, meaning that one year the field is soybeans and the next year is corn and the next year it's soybeans, and we keep going back and forth. There's benefits to doing this. The issue, though, of course, is that if you harvest the corn the one year, you can't collect every single kernel. Some of it's going to fall into the earth. In the following year, some of those corn kernels will voluntarily plant themselves, along with the soybeans that you planted. Now, in that case, then, while corn one year was something you wanted, the following year that corn is going to be taking the resources away from those soybeans. And so at that point, the corn is really a weed. And to try to kill off the corn in the middle of the growing season would be impossible if we used Roundup, right? You'd kill off the entire field, not just the corn, but also the soybeans. And so what they did was they genetically altered the soybeans to be Roundup ready. What this means is that the soybeans are resistant to the Roundup. You can spray them with Roundup and end up killing off the weeds even in the middle of the growing season. That's a huge advantage to being able to increase yield from your fields. At the same time, one of the things we might wonder is whether or not the amount of Roundup that's being used in the environment has increased or decreased or stayed the same, given that we can spray our fields whenever. Another major trait here is BT. BT, I know, does not stand for insect resistance, but this is the idea. BT in particular stands for a type of bacteria that lives in soil called Bacillus thuringiensis. And this particular soil bacterium makes a protein that is toxic to insect larvae. When the insect chow down on the bacteria, the toxin coats the lining of their intestines, prevents them from being able to absorb nutrients, and the larva ends up dying. In the United States, the European corn borer is a huge, huge pest. And as far as I know, it's present 2 times during the year, sometime in May and sometime in August. And a farmer would have to be vigilant against when that corn borer larva is present and then spray the field with the bacteria. That bacteria toxin, or the toxin that the bacteria produce, ends up being something that the larva would end up eating and then, of course, die. But if you're not vigilant, then what you would find is that the larva is chowing down on the inside of that corn stalk. And the next windstorm that comes by, the corn gets knocked down, would never get back up, and your corn crop is ruined. In addition, not only do you have to be vigilant, but you have to be able to spray, when it's not super windy, when it's not going to rain. And you can't do it too far in advance, otherwise the toxin by the bacteria aren't going to be out there. And so this becomes a bit of an issue. The genetically modified solution is to take the gene that the bacteria have for encoding that protein, put it into plasmids, transfer the plasmids into the corn plants, and now the corn plants can make that toxin, that bacterial toxin, every hour, every day of the week. To be clear, they're not making the bacteria, they're making the toxin. And again, we've been using this bacteria, the toxin from the bacteria, for, I think since the 30s or 40s. It's just now genetically engineered into the plant. We have to take a look at whether this is safe for the environment, whether this is something that has an effect on humans as well. But that's the intent of this BT trait. In the United States, there are some crops that are approved for GM. Corn and soy are huge on the list. There are others, including rice and sugar beets and tomatoes and squash. Approval does not necessarily mean that these crops are being grown and then distributed for food consumption, but the list keeps growing. I don't need you to have a comprehensive list, but I just want you to have an idea that there's a lot of different crops that are out there that potentially could be genetically modified. To show you this part here, this graph is showing us years, it's showing percent of planted acres, and it's showing us that since the advent of genetically modified crops in the mid to late 90s, that now, 20 some odd years later, the percentage of crops in the U.S. that are genetically modified, soybeans in particular, 95% of the U.S. soybean crop is genetically modified. Corn, somewhere around 80% at this point. Even cotton, I know not a food product, but cotton is also highly genetically modified. That's changed significantly since prior to the mid 90s. That's not just true here in the United States, that's true in many other countries around the world. South America countries, Australia, China, India. And this map has increased in the number of countries that are using genetically modified crops. And we'll talk at some point about African countries and European countries and some of the issues that they've faced along the way. Some traits that are there. I don't need you to know necessarily which plants. I do want you to have some familiarity with the different traits that are present. We've already talked about pest resistance and viral resistance and herbicide tolerance, but also drought resistance is a trait that's being introduced into corn plants. Increased nutritional value, and so golden rice with the vitamin A gene, heart healthy oils, so that when you're eating whatever it might be, that you're not getting saturated fat, but you're getting a variety of mono unsaturated fats. Improved fruit, so for processing and packaging, so that it survives better from the farm to the market. And then altered ripening, we'll talk about the flavor saver tomato at some point. Some issues that we'll have to take a look at in this unit. Some people are concerned about the creation of superweeds. The idea in this case isn't that the traits are migrating from the crops to the weeds, but certainly being able to spray your field with Roundup more frequently means that you're better able or sooner able to find those plants, those weeds that just so happen to have the traits that allow them to be resistant. You're speeding up the process of evolution in a sense. And so these super weeds, we'll have to take a look. Are they increasing in number? Have they developed more frequently? In fact, I'll show you some data here on that in just a moment. In fact, here's the data. And so what they're showing in this case, time, years on the x-axis, y-axis, number of resistant species. And here we are when genetically modified crops started to be introduced. And here we are with some that are genetically modified. In particular, glyphosate is the one that is the chemical that's found in Roundup. And you can see that starting in the mid-90s, all of a sudden we're starting to get an increase. And this means that once you find a plant that is resistant to the herbicide, then you don't have a way of getting rid of that weed. Now, this would have happened anyway, eventually, with the use of herbicides. But now that you can use herbicides more frequently, you're hastening the development, natural development of these superweeds. In addition to super weeds, you have the same idea with super pests. If the pesticide is out there 24 hours a day, 7 days a week, eventually you'll find those pests that are resistant. Some other issues, is it safe to eat? What about allergens that might be in there that are things that maybe people aren't aware of, that you're not in thinking that the food that you would eat would have the material that you're allergic to because you're not eating that particular food product. And then also killing unintended species, butterflies in particular, and we'll take a look at that issue through some readings. We'll also take a look at labeling. What are the requirements? How do we know if the food that we eat is genetically modified? And your assignment here, lab-wise, is to bring in a food item that, well, may or may not be genetically modified. And so what I'm going to suggest to you first is that you bring in something that contains soy or corn, and you'll find in the ingredients label lots of things with soy or corn. The best bet, though, is to bring in something that is less processed. So, for example, if you bring in something that is corn oil or soybean oil, or just has that as your soy or corn ingredient, by the time you get to the oil part, there's really not that many cells left, and what we need to analyze is the DNA. And so I wouldn't necessarily bring in something that is just has corn oil or soy oil in it, canola oil, but maybe bring in something that is a little bit less processed. For example, corn chips. If you're thinking about Doritos or Cheetos, if you're thinking about taco shells, if you're thinking about potato chips, well, I guess those would be potatoes, but a lot of things that you could bring in that are less processed. You don't have to bring in necessarily fresh corn or anything like that, but something along those lines. And you don't have to bring in a lot. A simple chip would be more than enough. You're going to weigh out a small little portion of this and process it, and so you'll have plenty of material available. Outside of that, we'll be working on some labs soon. If you have questions, let me know, and we'll see you in class at some point.